And welcome, everybody, to today's webinar with the title Novel Insights into the Role of Biomechanics in Cell Biology. We're excited to present today to the members of the American Society for Cell Biology. I believe with today's talks, we'll cover an interesting topic for you. The webinar will focus on the important role that mechanobiology plays in shaping our understanding of fundamental biological mechanisms. We will have two talks. First, from Professor Andreas Janzow from Göttingen University in Germany. Hello, Andreas.
Yeah, hello.
Thanks for joining. And Dr. Kimberly Stroka from University of Maryland in the United States. Hi, Kim.
Hello, everyone. Thank you.
Our speakers will give insights into their research and outline how atomic force microscopy, also known as AFM, an advanced multi-parametric imaging technique, can be used to study the nanomechanical properties of living cells, the morphology of brain endothelial cells, and the viscoelastic properties of epithelial cells. If you want to ask questions during the webinar, you can do so at any time in the Q&A panel. We will try to answer all of them either directly or in the chat or in the respective Q&A session directly after each talk. So without further ado, let me introduce our first speaker. Professor Janzow is a full professor at the University of Göttingen since 2008 at the Institute of Physical Chemistry. Interesting fact, this institute was founded by Walter Nernst and was the first one in Germany focusing purely on physical chemistry. His major interests are membrane biophysics, cell mechanics, sensor design, and single-molecule force spectroscopy. What I found most fascinating is that Professor Janzow and his team are working on bridging the length scales, researching samples from single molecules to tissues. The title of his talk is Viscoelastic Properties of Epithelia in 2D and 3D. Andreas, thanks for being here. Please go ahead.
All right, let me share the screen. All right, you should see the title right now. So welcome, everybody. The talk will be about viscoelastic properties in 2D and 3D, meaning that we are also trying at the end to cover a little bit our recent work on organoids, in this case, cysts. And I want to start with making clear that mechanical properties of cells are pivotal for many biological processes comprising cell motility, cell division, but also collective phenomena that involves many cells, like in epithelial to mesenchymal transition, wound healing, but also cancer and infection. And I want to start by showing you how single cells can be probed with an atomic force microscope in order to obtain parameters that are relevant for these processes. And this sketch shows you already the most important experiment we will do that involves indenture, that is a sharp tip, or also a blunt tip is used, attached to a cantilever that serves the force probe or force sensor. So the deflection of this soft spring is measured by shining a laser here on top and measuring the deflection of that cantilever gives us access to the force. This is shown here in this simple sketch a little bit in more detail. So you see again here the cantilever that comprises also the tip that is used not only for imaging, but also for manipulating cells. In this case, it's shown here as an indentation experiment. And this is the typical setup that is used by many manufacturers that employ these lasers that are used to read out the deflection that can be minuscule of these cantilevers. There's also an XY piezo that allows us to not only indent at a certain point, but also to image these cells and also to move up and down the base of these cantilevers. So when treating cells as objects of interest for mechanical properties, often the so-called Hertz model or contact model is used to describe the response of these cells. And it's a very old model. It is more than 100 years old, and it has been developed in a completely different context by Heinrich Hertz. And the beauty about it is that the force versus indentation relation is rather simple. So the actual response function that delivers us information about the elasticity is shown here. So it's just the elastic modulus, the so-called Young's modulus. Here we have the probe indenter size, basically probe size, and of course here the indentation depth to the power of 3 divided by 2. So depending on what kind of geometry, we have a little bit different relationships, but they are all very, very simple. So meaning that everything is lumped into one single parameter. We will see later on that this might not be sufficient. So if you do such an experiment, you see that the indentation here, this is force as a function of indentation, here's h, the indentation depth. And you see there's a nonlinear function here. And when you go back and relax the system to the initial state where you indented the cell, you see that there's a lot of energy dissipation taking place. And this actually tells you that the initial idea to treat the cell as a continuum might not be sufficient, but you need to implement somehow the energy dissipation here. This can be done readily by using the correspondence principle, the viscoelastic correspondence principle by Boltzmann, and then you can fit these curves rather nicely with the same physics behind the Hertz model of contact of 2 elastic, or in this case, viscoelastic bodies. So, however, there's a big problem here. And when we do that with different indenter geometries, we found that the elastic modulus here is a function actually of the indenter geometry, which shouldn't be. Of course, it's an intrinsic parameter, which is just a parameter describing the cell as an elastic or viscoelastic continuum. But it depends on the indenter geometry. Here you have a cone, and if you go to more blunter tips, you see that it's actually getting softer, which isn't possible. So there's something wrong here with these treatment of the cell as an elastic continuum, and everybody who's involved in cell biology knows that's a way more intricate system. A cell is composed of a membrane, and there's a cortex attached to it. There's cytoplasm, there's a crowded environment with lots of protein, there's a nucleus, and so on. So the question that arises is, what part of the cell is actually the main contributor to this response, which we measure with the atomic force microscope? And what is the origin of the fluidity, which we usually measure, this energy dissipation I showed you, and what is its value? And so we started off with looking at the cytoplasm first. So that's of course a candidate which might be responsible if it's really a crowded environment. There's of course lots of energy dissipation expected from the cytoplasm. So this couldn't be done by AFM, but we embedded here little beads that we tracked, and this Brownian motion in this complex fluid gives rise to what is called the generalized Stokes-Einstein equation that can be used to gather then the elastic or the shear moduli, the storage modulus, and the so-called loss modulus. And if we do so, what we get is we get these dots which you see here. This is the loss modulus, and this is the storage modulus, and it's orders of magnitude lower than the actual viscoelastic spectra we get by using the atomic force microscope. Here we used the force microscope and used distinct frequencies where we oscillated a little bit, only a few nanometers here in the cortex, and measured the response function. But that got G prime and G double prime. G double prime is here the transparent squares here, is actually showing us that's more or less an elastic contribution that dominates here over the energy dissipation, so the fluid behavior at lower frequencies. So the main take-home message here is that the cytoplasm doesn't contribute too much to our system here, system's response. All right, so the next candidate would be, of course, the shell. The shell is composed of a membrane, and there's neatly attached to this membrane via molecules, specific molecules like azrine, mosine, radixine, but also other molecules unknown or proteins that attach the cortex that's a thin layer of actomyosin, so it has contractile properties to the membrane. And if you take a look here, you see it has a thickness of maybe 100, 200 nanometers, but also a mesh size, and this is more important for the elastic contributions here, of maybe 15 to 200 nanometers. So from the mesh size and the thickness, you can compute the so-called area compressibility modulus, the 2D stretching modulus. And that actually tells you that it's around 0.01 Newton per meters. So that's very low. However, that might actually be a main contributor to our response function. But then we have to come up with a different theory. So we need to use shell theory to explain what's going on. So we still use a continuum theory here, meaning that we cast everything into an Hamiltonian that comprises here an bending contribution. So that's especially important if the cortex gets thicker and thicker. So bending depends very much on the thickness of the shell. And then we have a stretching contribution, so the lateral in-plane stretching. So if you deform a sphere, it's actually increasing its area and therefore generates restoring force. And of course here is the indenter force implemented. So if we have to make a decision here what kind of term is less important, you will figure out that since it's a very large radius here of the cell compared to its thickness of the cortex, that stretching dominates over bending by an order of 1000. That means that we can concentrate here on the effect of tension. And if we write tension now in a way that it's comprised here of the pre-stress due to the cortical contractility due to actomyosin acting here as a contractile unit, and a term which reminds you probably on a two-dimensional Hookean law. So this is the 2D elastic modulus, the area compressibility, which dominates the stretching response here. So if we put everything together, we have a model that we can use to describe our force indentation data and the relaxation data as well by implementing a viscoelastic relaxation here. So if you do everything which I just described, we get a formula here for the force as a function of the area dilatation. This is this alpha. This is nothing else than the change in area of the shell divided by its initial area. And if you do this and actually also implement here a power law for the energy dissipation and the pre-stress is here also implemented, we get this nice curve which shows you the force as a function of time now. Basically here you have the indentation to a certain threshold, then keep the distance between the tip and the surface constant and watch the force relax. And the relaxation here is due to this part here of the formula. And this actually gives us values for the pre-stress and the area compressibilities as a function of the different indenters. And it shows you now that there's no such trend now, for instance, softening with increasing indenter size, meaning that we have now a consistent theory in hand that we can use to describe the viscoelastic response of the cell towards indentation or stress. All right, so we are not finished yet, so we have to actually check what of the two contributions, the membrane or the actin cortex, is more responsible for the response. And so we came up with the idea to use giant liposomes as models for cells that are devoid of any cytoplasm, nucleus, and actin, so just the membrane, and use the tipless cantilever. So this cantilever has no tip. And so we compressed in this kind, you can see it here in the side view, you see here the giant liposome that is compressed here between the cantilever and the substrate. And by using the same theory I just showed you, where we threw away the bending contributions, we can actually describe the force as a function of compression depth curve and get an area compressibility modulus. That's basically identical to the physics of micropipette suction experiments you might be familiar with. So there's a big problem now. So we get 0.3 Newton per meters for the area compressibility modulus, which is extremely high. And this is due to the fact that the membrane is an inextensible material. It's a liquid crystal, essentially, that is very difficult to extend in XY direction because it breaks when you extend it by more than 3 to 5%. So it gives us a very high restoring force. And that's not what we find when we indent cells. And that's probably due to the fact that there's lots of membrane area stored in wrinkles and folds, and also in particularly in caveolae and microvilli and so on. So that means that upon indentation, the excess area is provided by the cell in order to diminish the actual stress on the membrane, which would be dangerous, of course, especially in epithelial cells. There's lots of osmotic pressure to be and mechanical pressure to avoid. And what you see is then that the apparent area compressibility depends here in the denominator on the excess area. So the more excess area there is, the less area compressibility moduli we find. And that actually explains why the cortex is more important in epithelial cells than the membrane, as long as we have enough excess area. If we take away the excess area, then we will face a different situation. However, before I come to this point, I want to show you the general law we found, and that has been also described by others before. That is that the fluidity parameter from viscoelastic relaxation, which is around 0.6 to even very close to 0, meaning more or less an elastic cell, is lying here on a line when we have a logarithmic dependency of beta depending on the area compressibility, meaning that dissipation is inherently linked to elasticity, and that actually is also clear if you look at eta, the loss tangent, whereas G double prime divided by G prime is the tangents of beta. So that means that we have only one trajectory along which we can change the mechanical properties of cell, and the cell can actually respond. So it can either change and get stiffer, but then it also gets less fluid. And there's only one way to change that, and that is by changing Ka independent of beta by changing the excess area of the cortex. And I'll show you how we can do that by manipulations. So what we can do, we use stretch the cells and thereby remove excess area. That's one way to modify this relationship. And you can see that we can shift these curves by increasing the size of the cells, shifting it to the right. So meaning that we get stiffer cells because more and more of the elastic modulus is reduced by, or is changed by the removal of the excess area. So by the removal of folds and so on, more and more the membrane actually takes over the elastic response and the cell gets stiffer. We can do the same by using a cyclodextrin that takes away the cholesterol from the bilayer of the plasma membrane. And what happens then is usually microvilli disappear and the roughness decreases quite tremendously. And there you can use the atomic force microscope for imaging purposes. So you can remove here these ruffles by using these methyl beta cyclodextrin that takes away the cholesterol, and after 3 hours you see already that it's getting very, very flat. And if you compare now the beta versus Ka relationship, then you see that it has the same impact than stretching. So meaning that by taking away the excess area of the cell, we make the cell stiffer. And that's one way that the cell can actually change its mechanical properties, not only along this trajectory, but also orthogonal to this trajectory. All right, so for part one, I want to summarize what we have learned, so we can see that as long as there's lots of excess area, there might be actually situations where the cytosol or cytoplasm takes over the elastic or viscoelastic response of the cell. However, most of the time we have excess area in intermediate regime, and the cortex has the dominant is a dominant feature here. And when we have very, very few excess area, then the membrane is eventually the stiffest part, which you would probably not expect from a 5 nanometer thick entity, that this is so important in cell mechanics. But in fact, it's way stiffer due to the fact it's so inextensible in 2D. So I want to switch gears now and go to a little bit larger length scale now, and that is using multiple cells. And I promised you I want to show some of our unpublished work here. And these are cysts that we generate from MDCK2 cells. These cysts can are comprising a volume that is not exchanging freely here with the surrounding, and it's a monolayer in 3D, if you want so. And we are interested in if we can use the atomic force microscope here also to measure the viscoelastic response of larger objects here. And for that purpose, we have glued very, very large beads of 20 micron size to a tipless cantilever and used that as an indenter. So it looks basically like this experiment here, shown on the left. You see that we can indent the cyst that comprises many cells, and thereby laterally extend it. And now we use a theory that has been recently developed by Xavier Crépin, who termed this superelasticity. And that means that the cell actually can respond to lateral stress by changing its shape and thereby generate or relax some of the stress. And if we also implement that in our theory, or in our shell theory here, by adding this superelasticity term, which you can see here, we actually can describe the force relaxation curves quite well. And this is actually shown here. We see here that the red curve describes the experimental data from the cyst, and you see here we have 20 nanonewtons of forces which we can apply to these rather stiff organoids here. And this is actually the values we get out. And one take-home message here is if you compare now the elastic moduli with the elastic modulus of a single cell, which is around 400 Pascal, we have now 10 kilopascal in for the lateral Young modulus, which is way higher. So meaning that these cells organized in cysts are extremely stiff. And with that, I would like to close. Thank the group who actually contributed to the work and you for your attention. I'm happy to take any questions. Thank you.
Yeah, thank you, Andreas. Very interesting talk. I think it gave a lot of insights to the fundamentals both and the challenges in working with the cells and organoids. It's like I said, really cool that you show this different length scales in your work. So there's a question from Igor Sokolov. The SEM images of the cell cortex you showed look like cell microridges, which can mechanically be quite different from the cortex, which is formed by the cross-linked F-actin. Do you think that the cell microridges can be considered as surface roughness and should be taken into account as a separate contributor?
Yeah, that's a very good question. Thanks, Igor, for raising that question. It's in fact, I think you actually published a paper on this brush part, which is taking part of the response at very low indentation depth, and I guess that's very important depending on the cell type. And this cell type we have used, the brush border is not that expressed, so that we need to take it into account, and we didn't find any reason for doing that, but there might be reasons where that is necessary, and it's a very important thing to actually look at what the cell looks like and adapt the theory to these situations. Yeah.
Okay, thank you. Another question is, there's a fundamental question about the viscoelastic measurements with the AFM. Are the samples in liquid medium or are they dry?
Oh, everything is in liquid medium, and in fact, they're all living. So it's unstained, and they're living cells. Yes.
Another question from Leonardo Martin is, would the cellular motor activity influence the viscoelasticity of the cytoplasm, cortex, or membrane?
Yeah, the motor activity is actually implemented in this model by having this pre-stress element in my theory. And this means that there is a particular contractility involved in the response. So you could say that's a pre-stressed system due to the action of motor proteins.
Thank you. Another question from Shatuan Singh, can't see the full name. When a sharp tip is used for indenting the cell, do you think that the cell viscoelastic response is local or comes from only the membrane?
Yeah, good question. In fact, I guess it's rather local, and most people who want to get a kind of averaging of the cell response use larger or blunter tips to average out these effects. Or you do mapping. I mean, with AFM, you can of course do mapping and cast everything into a 2D map of the cell's viscoelastic response. However, then theory gets a little bit more intricate because you do not indent in the center and cannot treat the cell as a spherical unit and so on. It's there are advantages and disadvantages of the local approach.
I think it also depends on the indentation depth, right? What you
definitely, yeah,
the contributor is in the end.
That's true. So we try not to indent too deep in order not to touch the nucleus and get a stiffer response from the nucleus, because it's known that the nucleus is maybe 10 times stiffer than the cell itself.
Yeah, okay. There are many more questions. Maybe one more. The SEM cell image shows like a porous next to the microvilli. Is it normal, and is this affect the measurement? And this affects the measurement, sorry.
Yeah, the SEM is, of course, of a dried sample. That's clear, I guess. And there might be also artifacts arising here. But what you see is, of course, not the membrane anymore. You see the mesh size of the actin, which is maybe 50 nanometers here, 100 nanometers. That's can be picked up by SEM.
Okay, thank you. Just a general comment for the audience. Please post your questions not in the chat, but in the Q&A panel. Then it makes us easier to answer them. But we will now move on. And Andreas, maybe you can have a look at the chat and answer some of the remaining questions. And yeah, I will introduce our next speaker. So Professor Kimberly Stroka is an associate professor in the Fischl Department of Bioengineering at the University of Maryland. She's the head of the Cell and Microenvironment Engineering Lab. Her research interests are quantitative cell mechanobiology, microfluidic devices for modeling physiological systems, as well as nanotechnology and live cell imaging. With the use of model systems, the aim of her lab is to understand the fundamental aspects of cell and tissue mechanobiology, with the long-term goal of advancing the scientific knowledge and directing new therapeutic strategies for diseases. The talk is titled Quantitative Assessment of Mechanical, Phenotypic, and Functional Heterogeneities in iPSC-derived brain endothelial cell monolayers. So thanks for joining. The stage is yours, Kim.
All right, thank you so much for that introduction. It's great to be here, and I'm excited to talk about my lab's work. So much of my lab's work currently focuses on the structure of the blood-brain barrier, which is a highly selective transport system that very selectively allows things into and out of the central nervous system from the vasculature. So your brain is made of a network of over 600 kilometers of very small capillaries, about 7 microns in diameter. And this structure called the blood-brain barrier is really comprised of brain endothelial cells that are linked by these tight junction proteins. There's other adapter cells that both physically and through secreted factors help to support that the brain endothelial cell tight junctions and kind of help link the cells together. And you can kind of see these SEM images of the tight junctions there. Now, there's this blood-brain barrier that's actually in its intact state can slow or prevent delivery of therapeutics into the brain. For example, in the case of maybe chemotherapeutics that you might want to get across the blood-brain barrier. So they can kind of present a delivery challenge, but also in many neurological diseases, there's a dysfunctional blood-brain barrier. So there's many questions surrounding why the blood-brain barrier breaks down in many of these neurological diseases. So we know if we kind of zoom in on these tight junction proteins, there's many specific proteins that are helping these cells link together. And both tight junction proteins, also adherence junction proteins are present and help maintain barrier function. We know that a protein called ZO1 or zonula occludens 1 links the actin cytoskeleton with other tight junction proteins that are actually homophilically binding the cells together. And also that ZO1 regulates the tension that's acting on weak adherin-based adherence junctions. So our hypothesis kind of early when we started this project was that cell contractility pathways would regulate ZO1 and maybe other junction protein presentation at the cell-to-cell junctions in the brain endothelium. So endothelial cells can sense and respond to multiple mechanical cues in vivo via mechanotransduction. This is well known. And my lab has been really interested in this effect of matrix stiffness on brain endothelial cell barrier function and function in general in mechanobiology. And so why matrix stiffness as our kind of cue that we're studying? First, it's a really important design parameter for blood-brain barrier models in vitro. And also matrix stiffness can be altered in disease, especially in the context of the brain in these various neurological diseases. So we have been using polyacrylamide gels that are coated with extracellular matrix proteins, and we're using gels kind of in the physiological range of stiffness. We use AFM to measure the Young's modulus of these gels. The gels are, like I said, coated with extracellular matrix proteins, and then we can add our endothelial cells, allow them to grow into monolayers, and then we have many questions about these cells and how they're growing on these various matrix stiffnesses. So one of our main questions most recently has been, what is the relationship between cell-to-cell junction phenotype, barrier function, and cell stiffness on a local level? So how are these things related? And can one kind of impact the other? And also, does matrix stiffness impact this relationship? So thinking about junction phenotype, local barrier function, and also cell stiffness as measured by AFM. So kind of thinking about junction phenotypes, as we look across literature and as you look at immunostained cells, cell-to-cell junctions can change in response to various mechanical cues or biochemical cues. And if you look across literature, there has been many qualitative ways to describe these changes, linear patterns, discontinuous, segmented, punctate, etc. And my lab kind of, as we looked at these immunostained cells of brain endothelial cells, we saw a lot of heterogeneity of these various types of junction phenotypes. And so we developed what we call the Junction Analyzer program, which is a Python-based image analysis software that categorizes the morphology of junctions around the perimeter of the cell. So we have specific definitions for what we consider to be a continuous junction, a perpendicular junction, or a punctate junction. It's really dependent on how much of the path length of that junction is within the cell perimeter. It has to be some minimum amount to be considered continuous. And then it's really the aspect ratio that determines whether it's perpendicular or punctate. So we have these definitions, and we have this mostly automated program that will then quantify the percentage of the cell perimeter that's taken up by each of these different junction phenotypes. So we've been using this quantitative method to really assess the junction phenotypes across brain endothelial cells in a range of different conditions. And one of the first things that we started out thinking about, kind of going back to this idea of matrix stiffness, we know that substrate stiffness can regulate cell contractility in endothelial cells. And so we were wondering whether cell contractility also influences ZO1 integrity in human brain microvascular endothelial cells, specifically thinking because ZO1 is that protein that is linked to the actin cytoskeleton and also links the homophilic cell-to-cell junctions. So we had hypothesized that in conditions of low contractility, either soft substrates, or if we add a biochemical inhibitor like a myosin 2 inhibitor, such as blebbistatin, or if we activate cyclic AMP, which leads to increased myosin light chain kinase, we hypothesized that these conditions would lead to more continuous ZO1 phenotypes and also better barrier function. Whereas if we put the cells on or kind of expose them to high contractility states, either through stiff substrates or by adding colliculin A, which is a drug that inhibits protein phosphatases. So, for example, myosin phosphatases, which leads to a net effect of increased cell contractility. We hypothesized that this would lead to more discontinuous ZO1 and poor barrier function. So we first looked at the effects of blebbistatin on ZO1 junctional coverage, and our results were right in line with what we hypothesized. So when we added blebbistatin alone, we saw an increase in monolayer coverage and also an increase in junction coverage, especially the continuous junction coverage. And then when we added the cyclic AMP activator, in addition to blebbistatin, we saw an added effect where, again, the continuous junctions were even more significantly higher in terms of their percentage around the cell perimeter, even in comparison with just blebbistatin alone. So there's an additive effect there. So then we put the cells onto soft substrates, kind of ranging from 194 kilopascal down to 1 kilopascal. And we saw that those, especially the 1 kilopascal substrate, promoted continuous ZO1 junctions, again, in line with our hypothesis that on these softer substrates, there's reduced cell contractility, and we see these increased percentages of continuous junction coverage. If we did the opposite, where we put the cells on the soft substrates, the 1 kilopascal substrate, but then we added the colliculin A, which again increases or inhibits protein phosphatases, including the myosin phosphatase, that induces contractility in the cells, and we saw the cells shift to a more immature ZO1 junction phenotype with a high amount of these punctate-like junctions. And we quantified that. If you look on the right over here, we saw that addition of colliculin on the soft substrate significantly reduced the continuous junction phenotypes, as well as, or if you compare that to glass, we saw no change in junction phenotypes with the addition of colliculin A, presumably because the cells were already very contractile on that stiff glass surface. And so further inducing contractility had no effect on the junction phenotypes. So this was all in line with what we had hypothesized in terms of the relationship between cell contractility and junction phenotypes. So there does seem to be this impact of matrix stiffness on junction phenotype in brain endothelial cells. Now, the next question we had was whether that had any functional effects. So then looking at those changes in junction phenotypes, is there any change in local barrier function in those regions of, for example, discontinuous junction phenotypes? So we have been using a local permeability assay, which has huge benefits over more global type barrier function assays like a Transwell permeability assay or a transendothelial electrical resistance measurement. Both of those just give you one number across an entire monolayer, whereas this local permeability assay allows us to vary in a high spatial manner, allow us to see exactly where the cells are permeable. So we have a biotinylated fibronectin ECM that we coat our surface with, and then we add our endothelial cells, allow them to grow into a monolayer, and then we add phytc-avidin above the cells and the culture media, and wherever that phytc-avidin goes through the cells, and this is over the course of just a couple minutes, so it's all paracellular transport that we are expecting to see between the junctions of the cells, and the phytc-avidin will irreversibly bind to that biotinylated fibronectin, and we can, at that point, fix and stain the cells and kind of compare where the junctions are and what the junction phenotypes are in relation to where we see regions of local permeability. So, for example, if we add the cyclic AMP activator to the cells, we essentially we're blocking myosin light chain phosphorylation with this addition of the cyclic AMP, leading to a lower cell contractility. And so when we do that, we see significantly decreased phytc-avidin binding, which is an indicator that the cells are less permeable. We see a significantly reduced permeable region area per image as well when we quantify that. So, and then, like I said, we can kind of correlate what the junction phenotypes are specifically around those local permeable regions. I won't go into all the details of that at this time, but I really wanted to get next into what we've been doing more recently, not just with primary brain endothelial cells, but with which I kind of just showed you all the data from our primary brain endothelial cells, but for induced pluripotent stem cell derived brain endothelial cells. It's really interesting because they seem to have a slightly different relationship in terms of their response to substrate stiffness. So we're using this differentiation protocol, differentiating induced pluripotent stem cells into brain endothelial cells following Dr. Ethan Littman's protocol. And so the first thing we noticed is that in these IBMECs, Claudin 5 junction integrity. So Claudin 5 is a specific tight junction protein in the brain endothelium. We see an increase in continuous junctions with increased substrate stiffness and a decrease in discontinuous junctions with increased substrate stiffness. So this is kind of an opposite relationship in comparison with what we saw with the primary brain endothelial cells. And the total junction coverage, you can see, also increases with increasing matrix stiffness there. So we were wondering if this translates to what we would expect for permeability of the cells. So we did the local permeability assay again for these cells and found that the regions, if we go back to, looking at the continuous junctions are, again, lower on the softer substrate. We also see the highest amount of permeability there, which would make sense because those discontinuous junctions are leading to worse barrier function, basically. And then when we looked via fluorescence microscopy, we saw increased permeable region area on those softest substrates for the IBMECs. So we're still trying to figure out kind of whether the IBMECs just have a different range of stiffness, matrix stiffness that they prefer and grow better on, or if it really is an opposite relationship in terms of their barrier function and junction integrity in response to matrix stiffness in comparison with the primary brain endothelial cells. But what we noticed that was really interesting was that looking across the permeable regions and noting where they specifically were in terms of the junctions and whether it's at a tricellular region, a bicellular region. So, where 3 cells come together versus where 2 cells come together or where multiple cells come together. We quantified that and found that the permeable regions were most often found at the tricellular junctions. We also stained the cells for tricellulin, which is a specific protein found at those tricellular junctions. And the IBMECs specifically do express tricellulin at those tricellular junctions. So these are true tricellular junctions. And so we were wondering, okay, so we do see this relationship between local barrier function and junction phenotype. And this holds for both the primary cells and the iPSC-derived brain endothelial cells, possibly with a different impact of matrix stiffness. That's kind of a puzzle we're still trying to figure out. But we were also wondering, thinking about those tricellular junctions and whether there's some relationship or impact of cell stiffness on the permeability of the cells and the barrier function. So we have been using atomic force microscopy to measure the stiffness of these brain endothelial cells. So we culture our brain endothelial cells actually in a plate, and we have, in some cases, we have astrocytes and parasites that we put in the top of the transwell. We actually remove that, but during the culture, they're able to secrete their factors, and the brain endothelial cells can form a good barrier function in response to those biochemical cues from the brain, from the parasites and astrocytes. But then we would remove that during atomic force microscopy, and now we are doing AFM imaging of our large sections of our cell monolayers. And Andreas gave a really beautiful introduction to atomic force microscopy and the quantitative aspects of that. So I'm not going to go into a lot of details, but we can generate these atomic force microscopy force curves, and during the approach, this indentation region, we can fit that to a model like the Hertz model and derive out the Young's modulus of the cells during that approach curve. And so we can derive these topological images of the cells and also Young's modulus maps. And in these Young's modulus maps, each pixel represents one AFM force curve. So this is a really cool way, using this quantitative imaging mode with the JPK NanoWizard AFM, we can get a really high resolution map of the endothelial cells and their stiffness across the monolayers. And so we did this across IBMECs. These are the iPSC-derived brain endothelial cells. The cells were plated on increased substrate stiffness as we go down across these samples here. So 1 kilopascal, 2.5 kilopascal, 15 kilopascal, and then 194 kilopascal. And so part of this project, we have been looking at the effects of matrix stiffness on the cell stiffness. And I'm not going to go into all the details of that right now. But what I will point out here is that we observed region-specific patterns of cell stiffness at these nice high-resolution AFM force maps. So, looking at these AFM stiffness maps, we can see really distinctly that the cell junctions are very different in terms of their Young's modulus compared to the body of the cell. And we're just indenting a few microns into the cell. So we are not hitting the nucleus. So the middle of the cell, we've actually noted to be the softest region, and the stiffer junctions of the cells, the junctions tend to be much stiffer. But we also noticed that if you go back over here, the tricellular regions actually appeared to be the stiffest. So kind of looking at this visually and qualitatively, even though the whole image is a quantitative map, but just kind of qualitatively looking at that, we noticed those like region-specific differences in stiffness. So we developed a MATLAB-based analysis pipeline to further quantify the Young's modulus of distinctive monolayer regions. So we looked at the bicellular regions, the tricellular regions, and also the cell body. And we kind of segmented out those various regions. And from that AFM image, we're able to extract the Young's modulus information from those 3 distinct regions. And so what we found is exactly what we saw qualitatively looking at these images, that the tricellular regions were the stiffest, and then the cell body was the softest. And that held true for all matrix stiffnesses that we looked at. We've also been treating or kind of co-culturing the endothelial cells with astrocytes and parasites. We see the same trend hold where the tricellular junctions are the stiffest, at least on the softer, we see them on the softer substrates. But also in general, treatment with astrocytes and parasites tends to decrease the cell stiffness in all regions. So I really just wanted to highlight here the utility of the JPK NanoWizard and this quantitative imaging mode, and how we can really nicely kind of start to correlate this kind of region-specific cell stiffness with what we see going on with the junction phenotypes and also local barrier function. We have seen that the tricellular junction regions are the stiffest and also the most permeable, and we think this could be due to high cell contractility at those specific locations. So, perhaps we can modulate mechanical signaling pathways or cell mechanical properties to strategically alter endothelial cell-to-cell junction integrity and maybe also blood-brain barrier permeability. So that's kind of the, I guess, the implication and relevance of why we're doing this. So with that, I would just like to thank all of you for coming today and listening to our talks. And definitely would love to thank my entire lab. Dr. Li Yan is the one who really did all of the AFM imaging and has been leading all of the IBMEC work. And then I'd also love to highlight Udit Gupta, who is an undergraduate student in our lab, who has developed the MATLAB analysis pipeline to kind of further quantify those AFM image maps. So with that, I will just stop, and I'll be happy to answer any questions.
Thanks, Kim. Very interesting. I found it quite surprising that different cell types respond differently to the substrate stiffness differences.
Yeah, and it's hard to say at this point whether it's a completely opposite effect or if the IBMECs just have a different stiffness that they prefer, like a different stiffness range that they prefer. It is known that even for peripheral endothelial cells and primary brain endothelial cells, that very soft, like if you go below 1 kilopascal, you're going to reach a substrate stiffness where the junctions don't form very well. So that just might happen at a higher stiffness for the IBMECs. Again, we're not sure what the reason is, but there's also concern that the IBMECs are slightly more epithelial in nature than endothelial. So that's kind of something that is a bit controversial in the field, I guess. But that could also be part of the reason we see the difference.
Well, I guess you won't run out of scientific questions to answer or to investigate anytime. Yes. So there are some questions from the audience. Yufai Xiao asks, did inhibiting myosin correlation with less TCJ localization?
Sorry, could you repeat that one more time?
Did inhibiting myosin, I guess, correlate with less TCJ localization?
Inhibiting myosin, we saw increased continuous junction phenotypes. So like if we treated the cells with blebistatin to decrease cell contractility, we saw an increase in the primary brain endothelial cells. We actually haven't done this with the IBMECs, but we saw an increase in the continuous junction phenotype, specifically of ZO1 in the primary brain endothelial cells.
I hope that answers the question. If not, please let us know. So Chatuan Singh Rajput asks, cells are cultured on the soft substrates. So in the AFM experiments, the measured force will have response from the soft surface along with the cell alone. How do you separate the response of the bottom matrix to extract the response only from the cells? There was a very similar question from Lisa Wilson for Andreas' talk, so maybe you both can jump in here about the contribution of the substrate stiffness, essentially.
To the cell stiffness. Yeah, we're only indenting just a couple microns into the cell. So if the cell is maybe 20 microns high, and if we're only indenting about 2 to 3 microns in, that's still very small compared to the total cell height. And so I don't think we have to worry about the effects of the matrix stiffness. But if we were indenting further, for sure, that would probably be something we'd have to be worried about.
Yeah, I guess, Andreas, for you,
maybe I would like to add that's actually also what we do. And since there's a constant volume of the cell, it's probably not so much affected by the substrate. However, I would like to recommend reading the papers of Ricardo Garcia, who has done quite some tremendous work on how to account for that substrate stiffness. And that can be done, in fact, and the theory can be extended in a way to account for the substrate stiffness.
Yeah, that's great. Thank you.
Thank you. Simon Bovio asks, hello, Kimberly, thanks for the interesting talk. About the AFM on the monolayer, do you know if the tip may be perturbed at the junctions because of the local topography?
Yeah, that's a good question. Maybe perturbed at the junctions. It's possible. I don't think we've seen anything specific to make us think that could be happening, but I'm not sure if we would see anything. So if anyone has any suggestions for how we might be able to determine that, that would be awesome. But yeah, I guess we haven't really thought about that. But it's indeed a good question.
So maybe I can add to this. What you can do is there are different channels in the AFM. So there's the force channel, right, that shows you the deflection in the z-axis. But there's also other channels like the lateral deflection of the cantilever that can tell you if the cantilever is bent sideways or so.
Yeah, okay, yeah, good idea, right.
So this is kind of a quick way to double check. If you just look at the force curves, look at the different channel and see if there's a signal there as well.
Okay, cool.
So another question, what would be the substrate's contribution when you compare the Young's modulus in different points of cells? So I guess it's related to the how high the cell is.
Yeah. Right. So I mean, presumably, yeah. I think I mean, if you look at the topographic maps, I mean, there is where are we here? It seems like the junctions are actually slightly higher than the cell body. Is that right? Yeah. When we go back to the topological maps. So there is some.
We don't see your.
Oh, sorry about that. Let me share my screen again here. Do I need to swap? Is that better?
Mm-hmm.
Yeah. If we look at the topological maps, we can see, there is some height differences, right, across the different regions of the cells. So the junctions are specifically brighter, which indicates a higher height here on the topological map.
Mm-hmm.
Yeah, so, but we're, yeah, that's a good question, though. It's just in relation to this isn't like the absolute height of the cell, though. It's just in relation to, because it's a whole monolayer, right? So our baseline, I think, is just the lowest point of the image. Maybe the JPK expert can tell me, but I think it's the baseline is the lowest point of the cell, right?
So usually there's flattening. So there's an average baseline, basically, over the entire image.
Yeah.
So it is comparable because the substrate is not going to be perfectly flat as well, or level with the scanner. So
right.
What you see is correct. Yeah, so you can compare the different junctions with each other.
Right.
To an extent, of course. Yeah. Okay, let's move on. There's another question from Den Cheng Hu. Hi, may I ask what type of AFM chips you used for multiple cell Young's modulus measurement, and under what imaging mode QI peak was in the JPK NanoVisit?
Oh yeah, so we are using their Bruker cantilevers. It's I can tell you SAA-SPH-1 micron. So they're cylindrical tips with a 1 micron bead. And sorry, what was the second part of the question?
Do you use those tips for all modes?
Yes.
For the QI and yeah, I mean, you use QI, right? This is
yeah, the QI, right? The quantitative image.
Because QI gives you the topography image and the mechanical information through the force curves at the same time.
Yep.
Yeah, so those probes are really good because they're hemispherical and they have a large tip, so you don't get artifacts from landing with the cantilever itself on other cells, but only with the tip. That is actually a good choice. Yeah, they're available in different radius of curvature. Another question is, I wonder how much does the tip cell geometry impact your mechanical mapping? I think we answered that in the talk from Andreas.
Yeah.
So,
yeah, and we haven't explored that specifically, but Andreas did a really great job of.
Yeah, but you chose basically the rounded spheres, which give you the best or the least error when modeling the force curves. Yeah, so the closest to the Young's modulus. And can you repeat the literature source for substrate influence on cell indentation? That is a question for. Did you mention the literature source?
Substrate influence.
I'm not sure if he means or she means.
What I think you said that.
Ricardo Garcia's work on nanoscale 2021, maybe. But Google will probably do the job.
Yeah, so too. Okay, I think our time is up. Thank you, everybody, for attending. Thanks for the speakers, Kim and Andreas. Thanks for the wonderful talks. Let me quickly make an announcement. No, wait. There's going to be the next webinar. Sorry, it's not a webinar, it's a workshop in collaboration with the City University of New York in New York City. It will be held from October 31st to November 3rd. You can attend this workshop free of charge and participate in a hands-on demonstration of the Bruker NanoVisit 5 BioAFM So if you want to see how those results can be obtained in live, you can go there if you're in the area. And with this, I want to conclude the webinar at this point. Thanks again, everybody. Thanks also to the organizers at ASCB, and have a good day, everyone.